I'm especially proud to present our next speaker, Professor Aaron Czchanover, laureate of the 2004 Nobel Prize in Chemistry. In addition to his numerous affiliation with many academies, Professor Czchanover is also the chairman of the Scientific Advisory Board of Rambam Medical Center. Well, it is now clear that the ubiquitin system discovered by Hershko, Czchanover, and Rose plays a major role in numerous cellular processes, and aberrations in the system underlie the pathogenesis of many diseases. Consequently, the system has become an important platform for drug development. Is it possible that drugs will be developed to cure all diseases? How much will it cost? Will our society be able to cope with it? Well, Professor Czchanover, it is my great pleasure to call upon you to answer these questions.
People have a dream, and the dream is that one day we are going to cure all diseases. We are going to stay young forever. This Time magazine cover will be materialized. By the way, this Time magazine cover came out about 15 years ago, and we are still far away from being there. And the question is the new personalized medicine going to be the magic solution? And what I want to do with you today is not to give you an answer, but rather to share with you some thoughts about where personalized medicine is coming from. Is it a new term? What is its historical sources? And where it is going to take us. Again, I think that people are looking for cure for diseases with physicians, with hospitals, with the medical establishment. But I think that the best way to cure diseases is to prevent them. And we are putting very little effort into preventive medicine, and we are rather trying to repair damage once it occurs and once it's irreversible. But if we look at most of the diseases today, many of the diseases are behavioral diseases. We are obese, we are eating non-healthy food, we are smoking. There are infectious diseases that we are bringing in many ways on ourselves. There are environmental diseases, and there are genetic diseases that maybe in the future, by understanding better the human genome, we shall be able to prevent. So I think that if medicine will shift mostly into preventive medicine rather than to curative medicine, we are going to avoid and to save much suffering in the world. But then I think we have to really change the set of our minds going from one way to another, and I'm not sure that society is ready for that. We are still relying much on traditional medicine, on physicians, hospitals, clinics, and so on and so forth. But let's go into the history of drugs, and the history of drugs is the history that leads us into personalized medicine, and the history of drugs is basically the history of drugs in the 20th century. There were many drugs before the 20th century. There were Greek medicine and Chinese medicine and Roman medicine, but it's only in the 20th century that we started to look at drugs in a very systematic way. Clinical experience, clinical experiments, thousands of people that are being tried on different drugs against placebo, against other drugs, the big pharma companies that were established only in this century. And let's divide it into basically 3 eras, and the third one will be that of personalized medicine. And again, it's my view, it has nothing to do with real history, but maybe there is some reflection of real history even there. So the first revolution that occurred between the 30s and the 60s was the revolution of era of serendipity. There was no science, people did not really look systematically into drugs, and I will bring you just one example, probably one of the most important examples of complete serendipity, and that's the discovery of antibiotics. In this case, it's penicillin. Sir Alexander Fleming, a microbiologist that worked on bacteria, saw that bacteria are growing, and then he left a dish on the table by serendipity, by mistake, left it open over the weekend, came after 3 days, and saw that a fungus was growing on this dish. But he noted also that there was a halo around the fungus, and around this halo, bacteria could not grow. Somebody else would have thrown this dish into the garbage, trashed it down, not Sir Alexander Fleming. He concluded that this bacteria, that this fungus, the penicillium, secreted a material, an antibiotic material, antibios, anti-life. He went on to isolate it and found penicillin. And we all know that antibiotics really revolutionized our life. The conclusion was not only that penicillin came to the world, but that fungus, in general, can secrete antibiotics. And then came Waxman and discovered streptomycin and so on and so forth, all based on serendipity. Alexander Fleming did not mean at all to discover penicillin, but via his good eyes as a scientist, via this narrow halo, a revolution came to the world. I will not bring you to other drugs. Aspirin, for example, also came to the world by complete serendipity, a personal treatment given by Felix Hoffmann to his father that was sick with rheumatoid arthritis. I will not go into it. But basically, major drugs that we are still using came to the world via complete serendipity, no scientific studies, systematic scientific studies at all. Let's go to the second revolution. And the second revolution harnessed the achievements in chemistry. People were able to synthesize basically every compound. There are a million chemical compounds. And then people decided to go into high throughput, brute force screening of large libraries of chemical compounds. You don't know the mechanism, you don't ask too many intelligent questions, you have cancer, you take numerous dishes, small dishes on which you grow cancer cells, you take zillion of compounds, you screen them, you get a lead, and then you take the lead, and out of many leads, at the end of the road, maybe one will become a compound. I will not talk to you about cancer, but I'll tell you about another blockbuster that was discovered by complete serendipity, by high throughput screening, and these are the statins, the HMG-CoA reductase inhibitors that are sold these days for $20 billion a year. And this is probably a magic drug, a truly magic drug that prevents second heart attacks, prevents accumulation of cholesterol in the endothelium of the coronary arteries. It came to the world by a screening of a Japanese excellent pharmacist, Akira Endo. He won the Lasker Award for this wonderful discovery. And this is the achievement of science, of chemistry, by having numerous compounds, and he screened them for cholesterol inhibitory drugs. This was not sufficient, and we are moving very quickly into the third revolution. And the third revolution is the revolution of what Lee Hood called the 4 Ps. One P is the personalized medicine, as we call it, but there are other Ps. It's called predictive, because by understanding the human genome, we are going to predict. It's calling the preventive, then I talked about preventive. We may be able, by predicting, also to prevent, because we are knowing that the person is going in 10, 20, 30 years to develop a certain disease like heart disease. We shall be able maybe to prevent it by modulating the genome, by pre-treating him, by manipulating his proteome or genome via many other ways, and it's going to be also participatory. The patient is going to take part in this process. So we are calling it the 4 Ps medicine. It's not just personalized medicine. And it's all there in the human genome, and I just pulled out 2 covers of Time magazine. And the secret for personalized medicine is in our human genome. But what was the need to develop this personalized medicine? Why didn't we settle down for the screens? What was not enough in the rapid screens, in the screens of millions of compounds that let us be satisfied? And why did we look for further developments? The problem was that current medicine, in many ways, is a one-size-fits-all. It's like a pajama. We don't have very many sizes of pajamas. We have small, medium, and large, and that basically will fit all of us. And the problem was that if we have a group of patients, let's say cancer patients, prostate cancer patients, breast cancer patients, and they look basically the same at the day of diagnosis, following 5 years, some of them separate. Let's take breast cancer patients that look the same at the day of diagnosis, or prostate cancer patients. 5 years later, some of them will die despite the treatment, and some of them will be completely cured. That means that from the very beginning, we did not diagnose them correctly. We put them under a too broad umbrella. And people realize now that even breast cancer, that is such a common disease, or prostate cancer, or many other diseases that are very common, are basically a huge collection of small group of rare diseases. They are not the same. If 2 women that enter the clinic behave differently, though at the initial day they were diagnosed under the same umbrella as having a breast lump of such and such grade, and think about the grade. What is the grade based on? It's based on microscopy. That is a 200 years old technique. There is nothing there. They are much more sophisticated. This drove us in order to understand the difference between these two women and these two men, and it brought us to the human genome. And one of the drugs that was based, obviously, on such a molecular diagnosis, we are already starting to see where molecular diagnosis is going to take us, is Herceptin. Herceptin is an anti-breast cancer drug, and it's based on a molecular diagnosis that only women that have the HER2, the mutated HER2 receptor on their breast tumor can be treated with Herceptin. Otherwise, it will be completely useless to treat them with Herceptin. So we start to understand it, but we still are far away from bringing it to the full range of differences between different people that are sick under the so-called apparent same diseases. So Herceptin is one example, and there are many other examples, but they are still far away from having, we are still far away from having the full landscape in front of our eyes. So here is the human genome, and the aim is obviously to sequence the genome of each and every one of us. When people are talking about the human genome, we are talking basically on the genome of one person, one individual, and we know his name, but that's not sufficient. And the Human Genome Project took about 10 years and cost many billions of dollars to sequence the single human genome. We are now approaching very quickly an era in which the sequence of a human genome will drop to below $1,000. An individual human genome, and will take about 15 to 20 minutes. So basically, it's going to be a regular lab test, a little bit more on the expensive side, but $1,000 is the price now of an MRI or a CAT scan or another sophisticated diagnostic examination. So this will reach the individual very quickly. People are predicting that it will be in our doorway within less than 5 years. The ability to sequence the entire individual human genome within few minutes and with a cost of less than $1,000. Now, based on the human genome, we shall be able to isolate the mutations of in woman A and woman B and woman C to diagnose them first, and then we shall be able to go via each of the steps and try to design drugs. So the roadmap is first diagnosis, understanding mechanisms, and only then designing drug. And if we are looking at the central dogma of biology, which is flow from the genetic material from DNA to RNA to proteins, which constitute our body, we shall be able to interfere in each step. We shall be able to do gene replacement or whatever. We shall be able to interfere with the RNA by siRNA and miRNA. For part of the audience, these terms look completely strange and out of the world, but we shall be able to interfere in each step, and once we shall reach to the proteins, we shall be able to design enzyme and protein replacement. So once we understand the differences between the two women and the two men, and we shall be able to approach them individually and to tailor treatment to each and every one of them. Well, it won't be treatment to woman A and B and C and D and so infinitely. We hope that at the end of the day we shall have groups and we shall be able to develop treatment to different groups. Well, the roadmap looks extremely easy, but is it going to be that easy? We already started the project. The Americans launched the Cancer Genome Atlas. They are taking now 500 cancers of each type, let it be colorectal, glioblastoma multiforme, pancreatic cancer. They decided to go to 500. The idea of 500 is that with 500 we shall basically know the information of 97% of all tumors. 3% will be outside with rare mutations. The cost of this 3% will be to increase the sampling significantly, and the price too, and the idea will be to go to 500 tumors, to sequence them, and then to subgroup them. Again, the roadmap looks extremely easy, and the Genome Atlas is already on the way. Well, now we started to think, is it really the way? Many diseases are multigenic, and the causative connection between the genes products is not clear, so we are going to be in a problem here. What will happen if we'll describe many of the genes? Who will be the chicken? Who will be the egg? Malignancies are characterized by genomic instability, and therefore targets are not stable. Let's say that we shall contain the cancer, then the cancer will escape by mutagenizing non-stop more and more genes. Human experimentation is very complicated. We are lacking faithful animal models. Cost of developing new drugs. Companies are liable to all kinds of lawsuits. Look at the case of Vioxx and Celebrex. How did it take the shares of Merck plummeting in one day? or companies don't want to go to new antibiotics. The antibiotic pipeline is drying out because antibiotics is amenable to development of sensitivity by bacteria all the time. Companies will not go into it. There are many bioethical problems with understanding the DNA. Who will reposit the information? What will be if this information will leak out? I'm just trying to point out to you very quickly that this is not such an easy solution as it appears on paper. And now, obviously, is the mutation. Without going into this wonderful map of what causes cancer, at the end of the day, as I told you, there will be, for each cancer, 10 or more driver mutations that are real causes, and then tens to 100,000 different passenger mutations in each cancer cell that started from the embryo, from the fertilized egg, and take us all the way to the cancer. And then at the end of the day, look at the green one, with chemotherapy, we are going to induce even more mutations. So the map looks extremely complicated, and people really doubt, some people really doubt where it is going to take us. And people already know. From the beginning of the cancer bank, we already know the tumors typically have from 30 to 80 single base mutations. So it's not one. We were dreaming on 1 or 2, but here comes 80. Except for types that take less time to develop, such as leukemia, because they are caused by environmental carcinogen. And then Fogelstein, who is one of the pioneers in the field these days, searched databases for all mutations in genes found in solid cancers in the past 2 decades for only 350 cancers, and he came up with 130,000 mutations in 3,000 genes. Again, the problem of hitchhiker from the real cause is still there, and what we are going to do about it. So people start to cast a doubt whether we are taking the right way. I'm not going to take you into the discussion. Just to point to you before I close up, one of the real pioneers in the field, Bob Weinberg from the Whitehead Institute, is really unsatisfied with this approach. He clearly says that we are going to achieve nothing by it. We are not going to increase the knowledge base. But since the sheet is very narrow, and we are going to pull the sheet out of the bed, and we are going to fund now this cancer bank, this cancer genome bank, we are going to pull out resources from basic research that drove science so far, and that takes me to some of the comments of Professor Hershkovitz before me. The sources of funds for science, we cannot have it all at the end of the day. If we could have it all, we could have gone and try everything and could be happy. And he is very much afraid that it's going to collapse the entire building on which science foundation stands. And he said that it's worse. Is it worth extinguishing, I'm reading here because at the second paragraph, 20 or 30 small-scale hypothesis-driven project to make room for an attack at a system-wide level? And he doubts it. And on top of it came the problems. The genome is not the source of our information, because beyond the genome, there is the epigenome. And beyond the epigenome, there is control by another level. And then proteins are subjected to many post-translational modifications that are not in the genome at all. We phosphorylate protein, we ubiquitinate protein, we glycosylate protein, we acetylate protein, and nothing out of that is going to be discovered by the human genome. So this adds another level of complexity, whether the genome indeed is going to solve this problem, and what way we are going to head, and whether personalized medicine in the way we dream about it is going to really solve the problem. And the people that are opposing this approach, and this will be my last slide, are saying, let's forget about cancer as an individual mutation or collection of mutation. Let's go to the main switch. Let's turn this main building switch off, and it will be better than dealing with the electricity in every single room and apartment. And they identified 6 different major switches that control cancer. One of them is that they evade apoptosis. They refuse to die. We bombard them with chemotherapy, with radiotherapy. They refuse to die. Any other cell will die. Let's turn on again the apoptosis switch. Let's go to the major switches. Let's go to the cell cycle. Let's go to the growth promoting factor. Let's forget about a single mutation or 2 or 20 mutations that drive cancer, and let's go to very sophisticated major switches, and they identify all of them, or those that are known to us, and they say, let's forget about the small wire, and let's take the major generator that feeds into the cancer cell and switch it off. So now, 10 years into the world of personalized medicine, into the time that the term was coined, we are taking a little bit back and trying to think about it again. And I think that we are into, I'm not going to offer you any solution. I'm not partying with any one of those opinions, but I think that as happens in medicine, once we become enthusiastic, we take back and think about the process. And at the end, I hope that scientists, smart scientists all over the world will come up with a wide solution. So thank you very much.